Hello everyone. I am Diva Whalen, a fourth-year PhD candidate at Meharry Medical College in Nashville, Tennessee, and I study triple-negative breast cancer. So let's begin from the beginning. Breast cancer. Breast cancer is one of those cancers commonly diagnosed in the United States, along with prostate cancer and lung cancer. Now, breast cancer is a cancer that is diagnosed mostly in women, and if we look at breast cancer in general, there are four main subtypes that are commonly talked about. You have luminal A, luminal B, HER2 positive and negative, and you also have triple negative breast cancer. Now, luminal A and B and HER2 positive, they have markers that are targetable or druggable. So therefore they have treatments, but triple negative breast cancer does not have any current treatments or direct targetable therapies for it. So that means we need to study triple negative breast cancer. And so in my lab, under the direction of Dr. Amos Sakwe, that is exactly what we do. We try to understand not only how we can discover a target for triple negative breast cancer, but what is the pathway or the underlying mechanism of triple negative breast cancer, and why is it so difficult to treat and also find a biomarker to treat and use drugs on. So in my case, my project focuses on a protein called RAS-GRF2. Now, RAS-GRF2 is a guanine nucleotide exchange factor, and what that all means is that RAS-GRF2 is a light switch for a oncogene called RAS. Now, oncogene is a protein that is associated with cancer behaviors, and the cancerous behaviors lead to cell growth, you have cell motility, and even metastasis or movement of that cancer to another location. And that's exactly what makes triple negative breast cancer so dangerous, is that if you have a cancer that already doesn't have a treatment and it moves to a different location, it becomes more difficult and harder to treat because it is in a new environment that it has to adjust to. Now, returning back to my protein of interest, RAS-GRF2. Now, RAS-GRF2 again works as a light switch to turn on and off the RAS protein. And this is very important because if you inhibit or turn off the RAS protein, then you will have a better chance of attenuating or slowing down the growth of triple negative breast cancer cells. So that means we will eventually, if or once my project pans out, have a biomarker that suggests that we can look at this pathway or this mechanism to target triple negative breast cancer. So why is this important? Again, triple negative breast cancer currently has no therapies used directly to treat triple negative breast cancer. We have drugs called chemotherapies, and these chemotherapeutic drugs, they work for women who have early onset of triple negative breast cancer. Yet, chemotherapy drugs also not only target cancerous cells, but it also targets healthy cells. So this is important because if we find a pathway or a mechanism or even a protein of interest or biomarker that signals for or that represents only triple negative breast cancer, then we can have a direct target for triple negative breast cancer and not affect healthy cells. So you won't have, hopefully, you won't have such severe physical effects on women or anyone with triple negative breast cancer that, that will be treated with the drug of interest. Again, I am Diva Whalen, and if you want to understand more about my work, I have a paper that was published in 2019 that talks about how RAS-GRF2 is indeed a crucial and targetable effector for triple negative breast cancer. Thank you.